Hello, everyone. I'm glad to introduce to you the research titled Spatio-temporal characteristics of the gastrointestinal resistome in a cow-to-calf model and its environmental dissemination in a dairy production system. Through multi-omics technologies, this work has revealed the distribution patterns of gastrointestinal antibiotic resistance genes in dairy cows, mother to offspring transmission patterns, as well as the environmental dissemination risks. The diffusion of antibiotic resistance genes ARGs in the environment has become a major health threat. As a natural reservoir of resistance genes, the gastrointestinal tract of ruminants, the mechanisms of vertical transmission from mother to offspring and horizontal transmission in the environment are still unclear. In this study, a cow-calf model was used to systematically analyze the spatio-temporal characteristics of the resistome, providing a basis for formulating prevention and control strategies. Rumen fluid and fecal samples from 136 dairy cows and 36 calves were collected from pastures in Gansu and Jiangxin provinces. Meanwhile, colostrum, meconium, and environmental samples were also collected. The dynamics of the resistome were systematically tracked through 16S sequencing, metagenomic, and metatranscriptomic technologies. First of all, this study found that there are significant spatial differences in the microbial composition of the rumen and fecal matter of dairy cows. The microbial diversity in the rumen is significantly higher than that in the hindgut, and the geographical region significantly affects the microbial community structure. Prevotella and Lactospiraceae are dominant in the, while the feces are rich in Ruminocacaceae, which lays the foundation for subsequent studies on the differences in resistance genes. The resistome analysis shows that resistance genes are dominant both in the rumen and feces. Samples from Gansu and Jiangxin exhibit region-specific resistance profiles. It is worth noting that the abundance of ARGs in the rumen is positively correlated with that in the feces, suggesting the flow of resistance genes between the gastrointestinal tracts. Through clustering analysis, this study identified three patterns of the resistome, all fecal samples clustered into one group, while rumen samples were divided into two groups. Prevotalaceae and Lactospiraceae are the key hosts of rumen resistance genes, while Ruminocacaceae dominates the fecal resistance genes, indicating that the microbial community structure shapes the distribution of the resistome. Tracking the resistome of calves from birth to 84 days reveals that the abundance of resistance genes in meconium is the lowest, but it reaches a peak 3 days after birth, with Enterobacteriaceae being the main host. As the age of the calves increases, the structure of the resistome tends to be stable, and the total abundance gradually decreases. Age are the main regulatory factors. The diversity and abundance of intestinal carbohydrate-active enzymes in calves increased with age. Glycoside hydrolases accounted for the highest percentage, and carbohydrate-active enzyme abundance was significantly and positively correlated with resistance gene abundance, suggesting a synergistic evolution of microbial metabolic function and resistance gene transmission. Resistome abundance and diversity were significantly higher in environmental sample soil, sewage than in biological samples meconium, colostrum. The co-occurrence network showed that mobile genetic elements MGES were the key vectors for the spread of resistance genes, whereas wastewater and soil were the main sources of dispersal of environmental resistome with the ecological risk scores. Metatranscriptomic analysis reveals that multi-drug resistance genes are dominant both at the DNA and RNA levels. Notably, ARGs like aminoglycosides exhibit higher abundance at the RNA level, indicating that low-abundance genes may exert a significant influence through high expression. This presents new challenges for risk assessment. In summary, this study reveals for the first time from the perspectives of time and space that the resistome in the gastrointestinal tract of dairy cows is geographically and intestinally specific, establishes a cow-calf model system to clarify the dual pathways of vertical transmission and environmental diffusion of antibiotic resistance genes ARGs in the dairy production system. The age and diet are the key factors affecting the early intestinal ARGs, and the water and soil in the pasture are the high-risk sources of ARGs diffusion. In the future, the risk of ARGs transmission can be reduced by regulating the early diet and optimizing environmental management. Thanks.